So for some reason, I don't think that the video came through today, so you can hear me talking. But I figured I'd put together something, just kind of a reiteration of what I said today with pictures here. I'll make this into a little movie. The first thing I did today was discuss a synopsis of the skeletal muscle physiology. Really simple. You have an action potential, which I've underlined once again here. Um, and this action potential travels down the axon of the somatic motor neuron, which is always myelinated by Schwann cells, reaches the terminal of Bouton right here. Calcium channels open by voltage. Acetylcholine is released always in the synapse between the somatic motor neuron and the skeletal muscle. Remember, the only things that somatic motor neurons or somatic efferents ever innervate are skeletal muscle. And the only thing that ever innervates skeletal muscle and elicits a flexion are somatic motor neurons. The autonomic nervous system never releases acetylcholine onto smooth muscles and causes them to flex. Just doesn't happen. In the skeletal muscle, nicotinic acetylcholine receptors, shown right here, are bound by acetylcholine. They open, sodium flows in. Its pathway through the cell is facilitated by these T tubules. In the T tubules, you have little DHP receptors, which are channels, as we'll see with cardiac muscle. That's a little foreshadowing, but you heard that today. But when these change configuration from this to, say, this, the top just flips open. When they pop open, no ions flow out of it. In the skeletal muscle, what happens is they have grabbed onto the ryanodine receptors and they pull those open. So you get a tiny little opening in the sarcoplasmic reticulum at the ryanodine receptors. Calcium flows out. Calcium then binds to troponin, specifically at the TNC subunit. This causes a change in configuration. This moves tropomyosin. Tropomyosin reveals the myosin binding site of actin, G-actin to be specific. Remember, G-actin are just little balls that are put in a line to make filamentous or F-actin. Myosin binds. When myosin binds to actin, it changes in configuration to where it flexes, causing a shortening of the sarcomere. And when this happens billions and billions of times over, you get muscle flexion. So that's this whole reaction. Then ATP has to come in and cause myosin to release actin. And then the myosin is stuck with ADP and an inorganic phosphate. Now, when myosin binds to actin again, right here, the ADP and the inorganic phosphate leave. So that's all of skeletal muscle physiology. Really, really simple, really easy. There's 3 ATPases that I want you to know. Look those up. Now, that brings us into cardiac physiology. And I'm going kind of quickly here because we had a conversation about it this morning. You can go back and listen to the audio. But basically, this is all the stuff I talked about this morning. I'm going to erase all these lines here. Cardiovascular system of humans here. Remember, we have a cardiovascular system because we're too big for diffusion. So we have to allow something to move through our body to carry all the nutrients, oxygen, pick up glucose that we eat, deliver it to all of our cells. And all of our cells, none of our cells are more than, I think it's 5 microns from the nearest capillary. That kind of shows you the innervation of all these capillaries in our body. There's nowhere in our body that's more than 5 micrometers. That's a 200th of a millimeter, which you can barely see. The heart causes, the reason that we have a heart is to form a pressure gradient. And this gets into flow and velocity and pressure. We'll talk about in just a moment. But you have a pressure formed here. That pressure then extends out through the aorta, through all of the arteries, the carotids, the brachiocephalic, the renals that go to our kidney, the gastric, the celiac that goes to our stomach, all those different arteries. The only pressure that they're getting is from the left ventricle pumping it out. Now we have 2 portal systems in our body, 2 major portal systems, the renal and the hepatic. Then there's a third one in the brain, the hypophyseal. And those are just special things where you have 1, 2 capillary beds in series before arteries give way to veins. Veins take deoxygenated blood back to the heart. Now, if we look at that over here, the kind of repercussion of that, when you have pressure, you're going to have flow. Flow is volume per time. So blood flow is the volume of blood that flows through the aorta at any given time. And to increase flow, you can increase pressure, or you can increase the diameter of your vessel. Increasing the diameter allows better flow. If you think about it, a milkshake. You take a little tiny coffee straw and you suck as hard as you can on that straw. How much milkshake is going to come through that little tiny coffee stirrer straw per minute? Not much. A few milliliters. If you have a garden hose, suck with the same pressure, a lot more milkshake is going to come through that, right? Which is why Freddy's gives you these huge straws so that you can drink their milkshakes. They're really viscous. Now, blood isn't very viscous. The viscosity of your blood really doesn't change under normal circumstances. If you're severely dehydrated, it might, but viscosity stays about the same, which is why I didn't really even mention it this morning. But what does change is the pressure that your heart sets up right here and the diameter of your blood vessels. Related to flow is velocity, and this is the speed at which blood's moving. Now, if the radius of your blood vessel stays the same, speed, flow, and velocity are really pretty constant. But you can increase the diameter like you're doing here of your blood vessel. That's going to increase flow and decrease velocity. So velocity and flow, when you're talking about the diameter of the vessel, are inversely related. If you increase pressure, you're going to increase both velocity and flow. That would be like sucking harder on the milkshake. You're going to increase the amount of volume of milkshake that's coming into your mouth. You're also going to increase the speed at which the milkshake is coming through the straw. Now, that's all relevant when you look at all of the different vessels in your body. At the aorta, the pressure is really, really high, and it goes down because as the cross-sectional area of your blood vessels increases as you go from the aorta to the capillaries, the individual capillaries are much narrower than the aorta. Say a millionth of the size, probably even smaller than that, but it's about a millionth the size. But there are many, 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 many more millions. There's billions of capillaries. So the pressure is going to fall pretty dramatically. cross-sectional surface area goes way up. That's what this black line is. Now, as you go to the veins, cross-sectional surface area goes back down, but you've already lost all the pressure. So it doesn't go back up. There's not a pump at your capillaries making the pressure go up. Now, how blood returns to your heart is because your skeletal muscle does serve as a pump, moving blood back through the veins to the heart. And once you get up into the abdomen, and then especially the thorax, breathing in creates a vacuum. That vacuum sucks blood up from the inferior vena cava into the chest, and then the recoil of the right ventricle is sucking blood from the atrium, from the right atrium, and that suction is sucking blood from the vena cava. So that's a little bit of background on the heart, more anatomy, but kind of vessel physiology. We're going to talk about vessel physiology a lot more when we talk about vessel physiology, when we talk about smooth muscle. So, cardiac muscle physiology here. We discussed this at length. So briefly, to reiterate, the initiation of the action potential in cardiac muscle is very different. You have no direct, meaning synaptic, neural control of the heart muscle cells. So there's no stimulus coming from the nervous system saying to beat. They are what we call autorhythmic. They simply depolarize completely on their own. There's no stimulus from the outside saying, hey, beat. We'll talk about the sinoatrial node in a lecture that I'll post later. But in essence, all of the cardiac myocytes, the ones that contract, all of them in the ventricles, they're going to beat on their own. And they do that because their membranes are leaky. And so they're going to leak until they reach threshold, which is about minus 80. And then it sparks an action potential in that cell, which then increases the increases the charge or decreases the voltage. It depolarizes the cell from negative 90, hits about negative 80. Sodium channels open, goes up to plus 20. Sodium channels close. There is an inactivation gate, just like in the neuron in these Hodgkin's-Huxley channels. Potassium channels open, bring it down. And then you have a calcium effect that we'll look at in just a moment. But that action potential causes these DHP-like channels to open. Here they're called L-type calcium channels, but they open, and instead of mechanically gating these ryanodine receptors in the sarcoplasmic reticulum, they let calcium in, and you get what is called calcium-induced calcium release, or CICR. Calcium flows in through DHP-like channels, causing ryanodine channels to open in the sarcoplasmic reticulum. Calcium binds to troponin, moves tropomyosin, revealing the myosin binding site on actin. Myosin attaches and flexes. Then ATP causes it to relax. Same exact thing that happens in skeletal muscle. Now, once it's done in skeletal muscle, all of the calcium goes back into the sarcoplasmic reticulum. In cardiac muscle, calcium ATPase is pumping like crazy to get the calcium back in, but you also have this guy right here, the NCX, or the sodium calcium exchanger, that pumps calcium out and sodium in. That sodium is coming down its concentration gradient, and you're using the energy stored in that concentration gradient to pump the calcium out. You get the sodium back out through the sodium-potassium ATPase. You can see all of that in a little flow chart right here. Now, the action potential is what causes those DHP channels in the T tubules of cardiac myocytes to open. Here we have the skeletal action potential, really short, 2 milliseconds. Cardiac muscle cell action potentials are a bit longer because here you have sodium coming in. Here you have potassium going out, just like in the skeletal muscle cell. However, here you have calcium channels that open. Calcium's going to flow into the cell. This is where you get that influx of calcium that you need the NCX to correct. Then calcium's going to remain open all along this plateau. Calcium channels close. Potassium channels open. And then bring the potential of the cell back to resting potential, which is about minus 90. Threshold is about minus 80, and they go up to around plus 20. Do I have that drawn anywhere? No. Now the blue line here, this is cardiac muscle contraction, and you can see it is pretty much the length of the action potential. Action potentials in cardiac myocytes, just like in neurons, have a refractory period. You can't start another action potential right here, right? You're not going to start an action potential right there. That doesn't happen. No, 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 no. It doesn't happen because it's got a refractory period. We talked about that. The kinetics of these proteins don't allow for another action potential to occur until here when it's done and the contraction's done. So by the time you're out of your action potential refractory period, the contraction's done. So what that ends up with is cardiac muscle cells cannot experience summation and they cannot experience tetanus. In the frog lab, you go over this whenever you do that. I'll have a little worksheet to turn in for the frog lab. Okay, that's about everything that I talked about today. It's kind of funny that I can do that in like 15 minutes if I'm not lecturing with you. I don't know how that works. But if you have any questions on that, I'm going to post this video. There's also all the audio on Teams. Sorry that it didn't post. I don't know what's going on there. Okay, great. I'll probably post 2 or 3 little 15, 20-minute videos so that you can take them in little bite-sized portions this weekend. Okay, see y'all Tuesday.